how animals are able to change aspects of their biology in different environments. And so we know that phenotypic plasticity allows animals to persist in different environments and allow them to increase their performance under varying environments. And so I'm really interested in what role microbes might play in aspects of phenotypic plasticity. And so the microbiome field is definitely exploding right now. And so another thing that my lab tries to do is sort of push the boundaries of this microbiome research. Right now the field is very dominated by studies that are comparing microbial communities across different environments. And my lab has done that as well. We've looked at the effects of temperature or captivity, environmental pollutants, fasting, diet, and sort of how these communities change in response to these variables. And I do think these studies are important. I always liken these studies to the early mammalogists that went out and had to study what mammals lived on what mountain ranges before we could come up with theories of biogeography and population theories and things like that. But we're specifically trying to push the boundaries of this field by, in one direction, what are sort of the functional consequences for these community changes to things like animal performance, ecology, and evolution. And then in the other direction, what are some of the molecular and physiological mechanisms associated with these microbe-dependent traits? And so as sort of a larger framework for my lab, I actually turned to a paper that came out as a result of a special session at a meeting of the Society for Integrated and Comparative Biology, where they aimed to identify the grand challenges in comparative physiology. And I sort of built upon those, followed up on them by adding a microbial perspective to these. And so these grand challenges would first be vertical integration of physiological processes across levels of biological organization. So we could think about things like how does gene expression influence how well a tissue is performing, how well does that tissue performance influence aspects of animal performance, and up to things like ecology and evolution. We can then add a microbial component. So the second challenge would be horizontal interactions of physiological processes across organisms. So how do microbes alter levels of gene expression or aspects of full animal performance, or even ecology and evolution, perhaps expanding dietary niches or determining whether an animal can act as a vector or a reservoir for potential pathogens. And then last would be temporal integration of these physiological processes over evolutionary change. So we could think about things like phenotypic flexibility, perhaps individual variation, and then even evolved differences across species. And so within this framework, today I'm going to present research on host microorganisms in 3 systems. So first we'll talk about sort of diet and nutrition and the rodent gut microbiome. Second, I'm going to focus on temperature and the tadpole gut microbiome. And then last, we'll talk just a little bit about how the gut microbiome might be used in species conservation. But we'll start with this first set, sort of diet and nutrition and the rodent gut microbiome. And so this work is primarily being conducted by a postdoc in my lab, Dr. Brian Tutteline. And he's asking questions like, how does diet composition alter animal physiology and the gut microbiome? Are these responses consistent across different species? And what role does the microbiome play in phenotypic plasticity? And so to test these questions, we've been using rodents as our study system. Rodents are the most diverse order of mammals, and they exhibit all sorts of distinct feeding strategies. So we have some herbivores down here, omnivores. Flying squirrels actually specialize on fungi, which have sort of unique nutritional challenges. And then we even have some insectivorous rodents over here. And so we want to try to understand how these distinct feeding strategies and distinct ecologies might structure host-microbe interactions. So as a first stab at this, we've conducted feeding trials with 3 species of rodents. So we focused on montane voles, which are herbivores, white-footed mice, which are omnivores, place these animals on these diets for 5 weeks, and then we are asking how does gut anatomy and physiology perhaps scale up to determine whole animal digestibility. So after this 5 weeks, we conducted a very detailed dissection of all of our animals. Each of these dissections took about 40 minutes with 2 people per animal. We did a total of 120 animals. So we took masses of all the organs, length, surface area, pH of all the gut regions. We saved tissues for enzyme analysis, gene expression. I think in the end we had over 3,000 tissues that are sitting in our freezer right now. And so I'll just present a few of our results. So the first one I'll present is small intestine length. And so when we look at our bowl, we actually don't see any diet effect. Sorry, let me walk you through this. So you're going to see a number of graphs like this where we have protein content on the x-axis, and then the color corresponds to the fiber content of that diet. And here we're showing small intestine length on the y-axis. And so we don't see any diet effect in our bowls. When we look at the white-footed mice, here we see a fiber effect, such that when these animals are fed a high-fiber diet, they lengthen their small intestine. We think this is an adaptation to sort of get more nutrients out of this challenging diet. But in our grasshopper mouse, we see a different response, that they lengthen their gut on a low-protein diet. And there too, we think that these animals are not adapted to low-protein diets, and so they may lengthen their gut in order to extract the most nitrogen out of the diet that they can. But here, I'd say the main takeaway from this slide and the view going forward is that we're seeing species-specific responses to these different diets. We can also look at something like cecum mass. So the cecum is the chamber in the hindgut where these animals are housing their fermenting microbial communities. And we see that dietary fiber increases the mass of this chamber. Here I'm presenting full cecum mass, but we have empty cecum mass, which is just the host contribution, and the patterns are the same. So in our bowl, we see that high fiber increases the mass of this chamber, again, likely an increase in these fermenting communities. In our white-footed mouse, we also see that fiber increases the size of this chamber, but the magnitude of this response is much larger, about 90% larger compared to 35%. And then in our grasshopper mouse, we actually didn't observe any change in cecal mass as a result of diet. So we can sort of look at these other organs, a diagram of the gut here. In our bowl, we saw that both protein and fiber increase the size of this foregut chamber. Protein increases the stomach size. Protein and fiber both increase pancreas size. But again, the major takeaway for now is that these species are exhibiting very specific, or I guess different responses to these dietary manipulations. And so I have a great team of undergrads that have been measuring all sorts of other aspects from these samples that we have collected. The students are working on histological measurements, measuring activities of digestive enzymes. We collected fecal samples, and so we can measure how well each animal was able to digest the fiber that we gave them. So measuring how much fiber was in their food as compared to the fecal pellets coming out the other side, and also nitrogen digestibility. And so I mentioned this idea of vertical integration, so understanding how things like gene expression or enzyme activity, things like that might scale up to whole animal performance. So like I said, we have measurements of digestibility in terms of fiber and nitrogen. And so once we've accumulated all these data points from all of these samples, our goal is to do path analysis to sort of understand what factors are driving aspects of whole animal performance. So here what you do is you sort of set up these hypothetical relationships between variables of interest. And so here we could think about things like gut size and enzyme expression might come together to determine total enzyme activity, and maybe that's what determines the ability of these animals to digest the food material. So this is still a work in progress. All right, so with these same samples, or with the same experiment, we were also asking, how do the microbial communities of these species respond? So we inventory the microbiome by sequencing the 16S gene. So the way this works is we start with an environmental sample with a mixed community of microbes, and each of these species has the 16S rRNA gene that we can use to identify a specific microbe. So we can extract all of the DNA from that sample, sort of amplify it and sequence that gene, and then what we get back are these really long lists of sequences that we can then identify two particular microbes and sort of infer the community composition of that original sample. And so we've done that with our samples from our rodents. So this is a principal coordinates plot, which is a very common way to present microbial data. And so here each point represents an individual animal, and points that are clustered together share more similar microbial communities. And so essentially what we're looking for are distinct clusters. And from this we can see that the species effect is very strong. So the grasshopper mice contain very different microbial communities than white-footed mice, and similar for the voles. We've had this data for a very long time. It's actually sort of saddening to me how long we've had this data and how little we've dug through it. So this is also still a work in progress, but we have done some preliminary analyses. Diet is also affecting the microbial community structure within each of these species. So species is a huge determinant, but then within there we are seeing variation based on diet. But we need to dig into that a little bit more. One thing we have looked at is just the diversity of the microbial community. So here we're measuring that by Faith's phylogenetic diversity. And across the board, we're seeing this protein by fiber effect. That when they're on a low protein diet, fiber tends to slightly decrease diversity, but when they're on a high protein diet, fiber tends to increase their microbial diversity. We've also conducted inventories of the fungal community. One critique that's very common in the microbiome field is that when we talk about the microbiome, we talk as if we're discussing the whole community, but our techniques tend to be very focused on only the bacterial community. And so we've been inventorying fungal diversity and also archaeal diversity. And here, at least for fungal diversity, we see a strong fiber effect in both our herbivore and our insectivore, that when these animals are fed high fiber diets, their fungal diversity increases. All right, so then we're also measuring aspects of microbial activity, especially in relation to fiber degradation. So when fiber comes in, it first has to be broken down by cellulase enzymes. So I have a student that's been measuring cellulase activity, and we see that high fiber diets increase total cellulase activity for all three species. No, not in the grasshopper mice. We're also measuring expression levels of transporters on the gut lining that bring short-chain fatty acids from the gut into the bloodstream. And so Ting Bei-Bo has been working on this, and she has found increases in these transporter expression levels, but only in our omnivore. And then we've also been measuring concentrations of short-chain fatty acids. So these are the products that microbes make from fiber degradation. And we're seeing a number of differences here, but I'm just going to present our data on propionic acid. So propionic acid is usually in the top 3 most abundant short-chain fatty acids. And what we were really interested in was that the grasshopper mice, we only detected this short-chain fatty acid in 3 out of our 40 individuals. And at first I didn't believe this. I had the metabolomics core clean their column, rerun our samples, and they still couldn't detect propionic acid in these samples. And so we think this is a true result and would be a massive difference in sort of the products of the microbiome between these species. So downstream, sort of getting overwhelmed by how much data we're creating without integrating it, but I promise we'll get there. And so the goal is to sort of combine aspects of host physiology with some of these measurements of microbiome structure or microbial activity to try to get at what factors are determining aspects of whole animal performance. All right. So like I said, the main takeaway from the results I presented so far are that these wild rodents are exhibiting species-specific responses to these diets. And so the question that I'm actually more excited about is asking whether these species-specific responses in physiology and plasticity are due to host genetics or whether the microbiome might play a role. And so we know that microbes can influence many aspects of host physiology, and there's a lot of interaction between host physiology and microbial metabolism. And one area that I think is really promising is the fact that short-chain fatty acids specifically have all sorts of effects on different aspects of physiology. And so where we saw that the grasshopper mice don't seem to be producing propionic acid, I think that could be a potential mechanism by which they're exhibiting these different physiological responses. And so we want to test this by using germ-free mice. You can almost think of these as bubble mice. They live in these sterile containers. They have absolutely no microbes in or on them. They're just completely bacteria-free. And so what we want to do is inoculate our microbial communities from our herbivore, our omnivore, and our insectivore into germ-free mice in order to hold the host constant and only change the microbial community. And so then the goal would be to run these sets of animals through these exact same diet experiments and ask whether we can recapitulate these species-specific responses to diet just by moving over the microbiome. Now, I don't think that we will completely recapitulate these responses, but I do think that demonstrating any role of the microbiome would be a significant advance in our understanding of phenotypic flexibility and animal physiology. So we're not quite there yet. So what we wanted to start with was asking, well, how well do these microbial communities even transfer over? And so we did a preliminary experiment just inoculating germ-free mice with our communities. However, this would have been a very expensive preliminary experiment if that was our only question. And so we followed this up with another question, asking, well, does the microbiome maybe influence diet selection? And so this is Brian Treveling, again, this time he's suited up. He's working with these germ-free mice that have now been inoculated with these very different microbial communities. So first they were allowed 1 week for the community to stabilize. And this has been shown to be long enough time for stabilization in other systems where they've moved sort of human microbes over to germ-free mice and things like that. And then we gave them 1 week of choice between a high carbohydrate diet and a high protein diet. So we can see that here we have sort of 2 little feeder hoods out the front of the cage. Each one contains either our high carbohydrate diet or our high protein diet. They were switched every day so that the animals couldn't form any sort of association with one side. And we just measured the amount of food that we put in and the amount of food that we can recover the next morning. And so a very common way to present measurements of diet choice or diet selection is using something called the geometric framework. And the way that this works is we would have protein eaten on the x-axis and carbohydrates eaten on the y-axis. And then we have these 2 rails here. And if animals were only given access to the high carbohydrate diet, this is sort of how or the trajectory that they would follow. They would be mostly increasing in carbohydrates, but increasing a little bit in protein. And similarly, if they were only eating on a high protein diet, they would sort of follow this line. And so this graph is hypothetical, but we can imagine that an animal first takes a meal from the high carbohydrate diet, it then eats a little bit from the high protein diet, decides to switch back, and eventually it's sort of moving towards this intake target within this space. And so that intake target is often what is compared when measuring aspects of diet preference. And so we've done this for our inoculated mice. Again, here the host species are all the same. It's only the microbes that have been introduced that vary. And we actually see significant differences between our 3 groups. So they're exhibiting different target intakes depending on what community they've been inoculated with. And we can sort of see this over time, that actually the herbivorous, we call them the herb mice, are actually eating more protein compared to the carnivore carn mice. At first this was sort of counterintuitive. I almost expected the carn mice to select a more protein-rich diet. But again, the gut microbes of herbivores are extremely nitrogen limited, and so it could be that they are manipulating host behavior for their own benefit. And we also know that herbivorous species exhibit a lot of protein hunger because the plant material that they regularly feed on is very low in nitrogen. So now we're trying to track down some potential mechanisms for how microbes might be controlling these behaviors. We then opened these animals up at the end of the experiment and measured some aspects of organ morphology. Now in this setup, we can't necessarily say whether the microbiome is directly affecting organ size or whether it's being manifest through behavior, sort of through this different diet selection. Because when these animals were being dissected, they had already been selecting independent diet compositions. But either way, we saw some very exciting results. So when we look at liver mass, we see a significant difference in liver size. And these mice have only been colonized for 2 weeks with these different communities. When we look at small intestine mass, we also see a significant difference. And then in some of these other regions, things like cecal mass and large intestine mass. And so I think this offers some really promising results for when we do our full feeding trial with these inoculated mice on these 4 different diet compositions. So in summary for this first section, it seems like these different rodent species exhibit varying responses to changes in nutrient composition. We're really excited about these results showing that the gut microbiome seems to contribute to diet selection. And then I think that the microbiome might contribute to these interspecies differences that we're observing in phenotypic flexibility. And so we'll move forward with those other germ-free experiments, hopefully this summer. All right, so now we'll move into the second section, looking at temperature and the tadpole gut microbiome. So there have been a few studies showing that temperature can influence the gut microbiome of ectotherms. So ectotherms don't regulate their body temperature, and so their microbes would be sort of exposed to whatever environmental or, I guess, body temperatures that those animals also exhibit. And so this project is being led by a graduate student, Samantha Fontaine. And I'll present just a few of her studies, first asking how quickly does temperature impact the gut microbiome. So like I said, there have been studies demonstrating changes, but often these were exposing animals to different temperatures for at least 3 weeks. And so we were curious, well, how quickly do these changes actually occur? And another question was, well, are these changes due to temperature because the temperature is directly affecting the microbiome, or could it be occurring through host physiology and affecting the microbiome? We still don't know that, but one way that we were trying to get at that is whether there might be any species differences in response to temperature. And then she is also conducting some studies asking sort of the reverse, whether microbes contribute to thermal performance. So we'll start with this first question. And so here Sam focused on 2 closely related species of amphibians, green frogs and American bullfrogs. And between these 2 species, they exhibit a great number of similarities. They're congeners, they have very similar morphology, similar habitat, and the individuals that I'll present were literally collected them from the exact same pond. They have the same reproductive phenology, they're herbivorous at the tadpole stage, and they share the same native range. Now one area that they differ quite a bit is how well they've been able to invade other ecosystems. So green frogs are largely restricted to their native range, whereas American bullfrogs are considered one of the worst invasive species and are present on every continent except Antarctica. And so we were sort of wondering whether whether the American bullfrog might exhibit a more plastic gut microbiome. So what Sam did is she collected wild green frog and bullfrog tadpoles, brought them into the lab, and sort of allowed them to acclimate at 24 degrees Celsius. She then split them into different treatment groups, either a group that was maintained at 24 or another that experienced a temperature increase to 29 degrees Celsius. And then she collected not repeated, but a series of samples at 12 hours, 24 hours, and then 2, 4, 7, and 10 days after this temperature shift. And from every animal, we sort of extracted the DNA and inventoried the gut microbiome of those individuals. So when we look at the green frog communities, here we see that these communities respond within 4 days of a temperature shift. So this graph is showing time on the x-axis and Gray-Curtis distance on the y-axis. It's not exactly the same, but you could think of this as the distance on one of those PCOA plots in terms of how different these microbial communities are. And so we can see that at 4 days, we start seeing this separation between our cool group and our warm treated group. Another way to look at this is actually with these PCOA plots. So at the 12-hour time point, our warm and our cool treated animals have a great amount of overlap. There's no distinct community structure. But at the 10-day time point, we sort of see this shift in community structure. There is still quite a bit of overlap. However, these results are statistically significant that the warm treated animals have a distinct community from the cool treated animals. Now, when we look at our bullfrog, here we actually see a response within 12 hours. This graph does not depict that, and I'm still trying to wrap my head around that. When we do these pairwise distances, there's quite a bit of overlap, but when we run Termanovas on these distance matrices, we do see significant differences starting right away at the 12-hour time point. So we can see that, again, in these PCOA plots, where even at the 12-hour time point, we're seeing distinct clustering between our cool and our warm animals. And here at the 10-day time point, they're not even overlapping. And so it seems like these bullfrog tadpoles respond faster and perhaps to a greater magnitude than the green frog tadpoles. And so we've also dug into some of this data in order to uncover which microbial taxa are responding. So in the green frog, we had 8 genera that respond to temperature and 1 that sort of shows this temperature by time interaction. And this would be this Clostridium species, where we see it sort of increasing in our cool animals, but maybe staying flat in the warm treated animals. And then if we look at our bullfrog tadpoles, here are some other genera. This Ray Ranella that seems to increase over time in our cool animals, and it stays as a flat line in our warm treated, and sort of the reverse for this Legionella. And so overall, it seems like temperature rapidly affects the ectotherm gut microbiome and in a host-specific fashion. So green frog tadpoles, we don't see a significant response until about 4 days after this temperature shift. Whereas in our bullfrog tadpoles, we observed a significant difference even at the 12-hour time point. And so in the future, we're really interested in studying perhaps what are the physiological consequences of these microbiome shifts. What aspects of host physiology might be driving the microbiome's response to temperature? So this could be things like the immune system or mucus on the gut lining that could be influencing what microbes reside in the gut. And then I mean, this is sort of a far out question, but sort of whether these plastic gut microbial communities might contribute to the ability of bullfrogs to exhibit this massive amount of range expansion, be such a dominant invasive species. So the second experiment from Sam's set of results is now sort of asking the flip side of this. Whether microbes contribute to aspects of thermal performance by the animal. And so specifically, she's been rearing tadpoles in regular pond water, which would contain a very diverse microbial community, or we can autoclave that pond water in order to sterilize it. And then she's been specifically focusing for now on aspects of thermal tolerance and also thermal acclimation. And so for this study, she is focusing on green frogs. And she went down to Louisiana to get wild caught individuals. We went further down south to try to get animals that are living in a warmer climate. And so what she did is she went down, collected the frogs, also collected pond water from their local environment, and then drove it all the way back to Pennsylvania. Although fieldwork sometimes involves things like this. This is the benefit of being a PI. I didn't have to deal with this. I only had to deal with phone calls about being stuck in the mud out in the middle of nowhere. But eventually she made it back. She kept all her pond water on ice. Drove very quickly back to Pennsylvania and got it in a cold room. And then she induced spawning of these adult frogs in the laboratory. So we wanted to rear our tadpoles in the lab so that we could really control what types of microbes they were being exposed to. And so after the sorry, it's sort of hard to see, but there are some egg masses here. And then once she had tadpoles, she put them into containers with either regular pond water or autoclaved